Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatrin Sawadi. And today I'm going to discuss about the prokaryotic and eukaryotic ribosome. I have discussed in detail in first part of this lecture about the structure, discovery, and many more related to the ribosome, chemical composition and functions, and many more. So today I am just going to share about the prokaryotic and eukaryotic because most of the students are not familiar with the prokaryotic and eukaryotic ribosome differences. They are just knowing that one is the 70S and other is the 80S, but today I am going to share each and everything related to the prokaryotic and eukaryotic ribosome. As you are familiar that the eukaryotic and the prokaryotic cell have many differences. Like prokaryotic cells don't have any membrane bounded organelle, means the mitochondria, nucleus, Golgi bodies, chloroplast, endoplasmic reticulum, and many more. But the eukaryotic cells have all the organelles like mitochondria, Golgi, all the membrane bounded organelles. But there's just one small granule-like substance or the organ, which is the ribosome. Ribosome present in both prokaryote and eukaryotic cell. In eukaryotic cell, ribosome found directly in the cytoplasm as well as in attached form to the endoplasmic reticulum for the protein synthesis. But in case of the prokaryote, ribosomes are just present in the cytoplasm because prokaryotic cell lack the endoplasmic reticulum, so this is directly, and you are familiar with the structure of the prokaryotic cell and eukaryotic cell. If you don't know about this, must visit to my lecture about the prokaryotic and eukaryotic cell. Okay. Now, as we are familiar with the structure of the ribosome consists of the two subunits, larger subunit and the smaller subunit. And the ribosomes are mentioned with the S, like the 70S and the 80S. And I have discussed in my previous lecture that S is stand for the sedimentation rate. You say that this is the Svedberg unit because it was discovered by the Svedberg, that's why S. Is it clear? So chemically, if you're talking about the ribosomes are made up of the proteins and the ribosomal RNA in the nucleolus of the cell. Is it clear? So, larger subunit and smaller subunits are different in the prokaryotic and eukaryotic, which I am going to discuss here. As I have discussed the structure in detail, that the two subunits attach with each other with the help of the magnesium ion, and there is a site for the messenger RNA, and this is the site for the attachment of the transfer RNA and for picking amino acids from the cytoplasm, and then attached here enter from this site, and then here the polypeptide chain is formed, and then at the exit side what will happen, the transfer RNA leave the ribosome. So this is all about the structure as well as a little bit about the function of the ribosome. Okay, now prokaryotic ribosome. First of all, I'm going to share prokaryotic ribosome. Prokaryotes have 70S ribosome respectively. Subunits comprising the smaller or the little subunit of 30S and the bigger or the larger subunit of the 50S. And as you know that why we are not adding the 30S and 50S, because this is not related to the size, this is the sedimentation rate. So it becomes 70S. Is it clear? The smaller subunit has 16S ribosomal RNA subunit. Must keep it in your mind. As we know that the ribosomes are made up of proteins and the ribosomal RNA. Is it clear? In the nucleolus of the cell. So the smaller subunit, which is 30S, it is made up of 16S RNA. And this RNA is about 1540 nucleotide. Is it clear? That bound to the 21 protein. Means 21 protein and 16S RNA make the smaller subunit, 30S subunit. And the largest subunit is composed of how many? 5S RNA subunit, which is about 120 nucleotide, and 23S RNA subunit, which is about 2900 nucleotides, and 31 proteins. Is it clear? So the smaller subunit is made up of what? This is made up of 16S RNA, and the largest subunit is made up of 5S and 20S RNA along with the proteins. And how many proteins are present in the smaller subunit? There are 21 proteins. Must keep it in your mind. And 31 proteins in present in what? In the large subunit. You can make your MCQs by using my lecture too, and also your notes. Okay, so must watch and till the end, and also note down the lecture. Okay, now I'm going to discuss about the and showing the structure of the eukaryotic ribosome. So eukaryotes have 80S ribosomes respectively. This is comprising of the small subunit of the 40S and the largest subunit of the 60S. Is it clear? So the smaller 40S ribosomal subunit is prolate ellipsoid in shape and consists of one molecule of 18S ribosomal RNA. Is it clear? And 30 proteins. And proteins are mentioned in S1, S2, S3 and so on. Is it clear? So 40S is made up of 18S ribosomal RNA and 30 proteins. The largest subunit, which is rounded in shape and contain a channel through which growing polypeptide chain make its exit. As I have shown you here, this is the exit site. Is it clear? Okay. Now the largest subunit, which is 60S, made up of 20S ribosomal RNA, 5.8, 3 types of ribosomal RNA. 28S, 5.8S and 5S. And how many proteins? 40 proteins. 30 proteins are present in smaller subunit, but the 40 proteins are present in the larger subunit. And how many types of ribosomal RNA? 3. 28S, 5.8S and 5S. But in 40S smaller subunit, just one ribosomal RNA, which is 18S ribosomal RNA. Is it clear? You can see here. This is the 80s. This is made up of the 3 types of the ribosomal RNA, means different sizes. These 5S, 5.8S, these different sizes of the ribosomal RNA. 5S, 5.8S and 20S RNA. This is just in prokaryotes and eukaryotes, you can see the differences. Is it clear? Okay, this is 16S and this is 18S RNA. And I have also given you the concept and also the amount of RNAs in proteins in the subunits of the ribosomes. I hope so the lecture will be clear upon you. If you have any question, kindly write in the comment box, and I will be back with my new lecture inshallah soon. Till then, Allah Hafiz.